Right, so we're going to be talking about histamine and antihistamine, so getting both sides of the inflammatory mediator that our body produces and then also the pharmaceutical that we would administer to counteract those effects. So it's going to be divided into those two halves. The first part talking about the structure of histamine on a sort of biochemical level, then talking about some of its receptors and the functions of histamine in the human body. And then the second part will be talking about H1 antihistamines as a sort of case study, partly because there's really not as much known about H2 antihistamines, and H3 and H4 antihistamines don't really exist. So that's how this is sort of going to go. We'll talk about a little bit about their pharmacological properties, therapeutic uses, and some of the side effects as well. So histamine was first isolated and characterized around a century ago, and basically it was noticed that it was able to stimulate smooth muscle and induce all of these vasoactive properties. And people started drawing connections between these vasoactive properties and anaphylaxis, which obviously is associated with things like, mass leukocyte infiltration, vasodilation, and things like that. It's predominantly made by mast cells and basophils, but you do see it produced from other cells as well, and it acts on quite a host of cells, and we'll get into a lot of these later on. And because it was first isolated from the tissue, not from the blood, it was given the name histamine, coming from the Greek histos, which if you can think of words like histology, for example. So it is synthesized through the amino acid L-histidine, and it completely relies on this one enzyme called histidine decarboxylase, or HDC. So without this enzyme, the human body cannot make histamine. That should already sort of be ringing some bells for you that this might be a potential target therapeutically. As far as metabolism goes, you can look on this image on the right, because around 97% of the histamine in our body is going to be metabolized through these two pathways. Now, the pathway that we're more interested in is the one that goes through histamine N-methyltransferase, and this turns histamine, as the name suggests, into N-methylhistamine, and this is converted later into a urinary metabolite. And that's relevant because when we test for bodily and physiological levels of histamine, we're not actually most of the time looking at the blood, because those blood tests are going to take a lot longer. We're looking at the urinary metabolite. So we don't actually measure directly histamine. We measure N-methylhistamine, which is a metabolite. And that's just directly related to how it's metabolized in the body. So let's talk a little bit about the enzyme itself. You can see the reaction here on the right. It's a decarboxylation reaction. Being the only source of histamine in the body, it's not surprising that when we want to regulate histamine levels, we're almost always going to do it at the level of the enzyme or at the level of the transcription of the enzyme. So it's very responsive to various cytokines that will either increase or decrease its activity. So for example, something like IL-3, IL-12 might increase production of histamine through this reaction. And something to note is that even with cells that don't natively store histamine, so for example, mast cells store histamine in their granule components, they don't have to synthesize them de novo every time they degranulate. A lot of cells do, and we can sort of observe that by looking at levels of the HDC enzyme. And this is actually how we know that, or how we first found that there were bacteria that were able to produce this enzyme. And something else that's interesting to note is that when we do knockout experiments and we take out the HDC enzyme in mice, for example, the mice have significantly lower levels of mast cells and decreased robustness of their granule components. So again, what that suggests is that this is not purely a synthesis enzyme, but rather this enzyme or perhaps its downstream components are actually directly linked to mast cell survivability in the body as well. Something that we've talked about many times is why haven't we evolved beyond these molecules, right? So we know they're responsible for pretty adverse effects, and one of the reasons could just be that nowadays allergies and atopic conditions aren't really quite fatal enough to sort of push us in any sort of evolutionary direction. But the other reason is that histamine is actually quite beneficial in the human body. It just works in ways that aren't, are typically overshadowed by its other roles. So it does things like promote cell proliferation, hematopoiesis, helps with embryonic development, wound healing is a really big one. And something that surprises a lot of people is that it's actually quite a big molecule in the central nervous system. So it's very central to our sleeping-waking cycle. It's involved with memory and cognition. And we'll see that its role in the central nervous system and the fact that it has receptors in the central nervous system is actually really important to the distinction between first and second gen antihistamines as well. And another really important thing to talk about is the fact that in experiments where we knock out the histamine decarboxylase enzyme, not only do we see lower rates of mast cells, but we also see increased rates of colon and skin cancer. So the working theory there is that there's some role of histamine that is involved with early cancer development, or more accurately suppressing it, that is sort of revealed when we do knockout experiments. So there are 4 receptors, and they're all GPCR receptors, and they're notated in a few different ways. You might see them listed as like an H1, H2. You might see them listed as H1R, H2R, or HR1, HR2, and they all mean the same thing. And we'll dive into each of these receptors, and how they're different from each other. But really, the big ones that we want to focus on are the H1 receptors and the H2 receptors. And there is a little bit that we know about the H3 and the H4, but not quite as much. And we certainly don't have any sort of therapeutic drugs that are working on those that are approved for consumer usage right now. And something that I'll mention right now before we get into the mechanism of antihistamines later is that histamine receptors generally are going to be constitutively activated, meaning that they are able to sort of induce these downstream effects even if the histamine ligand is not actively occupying the receptor. And so the way that these receptors work is that they're not in an equilibrium state between being occupied and unoccupied, but rather they're in an equilibrium between their active and inactive states, and we'll see how that's relevant to antihistamines later. So starting off with the H1 receptor, we'll see that with a lot of these receptor overviews, they're expressed on a lot of very similar cells. So I went ahead and highlighted some that I thought were particularly important. But the histamine receptor is obviously going to be, as I said, on the central nervous system. We see it on a lot of smooth muscle cells, which makes sense because that's where most of the allergic symptoms are coming from. You see it on eosinophils, lymphocytes, leukocytes, all that sort of fun stuff. So the pathologies that the H1 receptor is most involved in are going to be allergic rhinitis, urticaria, most of the allergic diseases, but we'll get into what that really means later on, and then some neurodegenerative and central nervous system diseases as well. And just for some notes about how it works, so we know from the IgE lecture that it's really dependent on calcium signaling. And we know that because histamine working on the H1 receptor is going to result in things like vasodilation, it's not surprising that nitric oxide or arachidonic acid are also going to be implicated there. Now, the transcription factor that I want to associate with the histamine receptor the most is going to be the NF-κB transcription factor, and that's something we talked about earlier. So keep that in mind as we move on to the mechanisms later on. Another thing that we see when we knock out the H1 receptor is that we get various neurologic deficits. So memory, locomotion, learning, all of these functions sort of get impaired when we don't have this receptor. So again, it's a really big function of histamine in the central nervous system is to act on these receptors. As far as the H2 receptor goes, the way I would think about this one is the cell I really want to highlight is the gastric parietal cells, and that's not surprising because the main pathologies this is involved in is going to be peptic ulcer disease and GERD, or gastroesophageal reflux disease. But the real interesting thing about H2 is this potential immunoregulatory role that it has. So we see that on non-mast cells and non-basophils, the H2 receptor actually serves as a sort of negative feedback mechanism that allows the body to keep histamine in check rather than just constantly sending out these signals. So H1 and H2 are sort of different in that one is much more inflammatory, whereas this one is slightly anti-inflammatory in certain contexts. The H3 receptor is going to be one of the ways that we sort of link mast cells and neurons. So we can think of the mast cells and neurons existing on a communication axis that is mediated by these H3 receptors. So it's unsurprising that we would see H3 receptors implicated in a lot of neurologic disorders such as Alzheimer's, ADHD, schizophrenia, epilepsy, that sort of thing. And I will mention that there is a possible negative feedback mechanism at work here as well, but this is less researched. And again, it's very context dependent, so a little bit unclear at the moment. So the H4 receptor is the last one to be formally characterized. This one we know the least about. We know that it's present on all of these cells over here. I won't waste time going over them. Potentially involved in some of these main atopic conditions as well. But the interesting thing is that there's a pretty high affinity with these receptors, or cross affinity with H1 antihistamines. So potentially, if someone's not entirely responsive to H1 antihistamines, that might be relevant to, I guess, the H4 antihistamines, and we are working on some H4 antagonists at the moment as well. So let's talk a little bit about histamine and some various cells. So the dendritic cells are a cell that are sort of surprisingly implicated with histamine. So it has a pretty big role in their differentiation. During dendritic cell differentiation, they produce a lot of endogenous histamine, and this acts sort of in a paracrine or autocrine fashion, and it starts to modify these dendritic cell markers towards their more mature state. And what's interesting is that all four receptors are expressed on dendritic cells, but they have different functions. So, for example, H1 and H3 are going to be positive stimulatory signals that encourage antigen presentation and encourage the production of pro-inflammatory cytokines. Whereas on the other hand, H2, as I sort of suggested earlier, is going to be an immunosuppressive receptor here. So these will be responsive or have more of an influence on decreasing antigen presentation, decreasing ICAM1 expression, and increasing IL-10 production, which all are going to be ways that we can sort of regulate the immune system. Now talking a little bit about T cells. So Th1 cells are going to be mostly expressing the H1 receptor, and Th2 cells, they express a lot of H1, but also a little bit of H2. And so this is a interesting point to touch on, because this is one of the ways that Th2 cells can sort of be involved in regulation of allergic reactions as well. So there is some sort of checks in place to keep this from going overboard. And on Treg cells, those cells that are supposed to tamp down the immune system, H1 can promote IL-10 production as well. So one receptor having multiple effects across different cells. And there is some suggestion that stimulating the H4 receptor will cause increases in FOX P3 positive Treg cells, and these are the cells that are going to be responsible for, again, toning down the immune system. So really all of this is to say that the role of histamine and the role of histamine receptors is going to be a little bit more nuanced than you might typically see it in a broad survey of allergy immunology, which is just to say that histamine causes all of these negative symptoms and antihistamines are going to shut all of those off. The final thing I want to touch about in this histamine lecture is just talking about bacteria. So this is sort of a very new thing that is being introduced to the world of allergy immunology. But the gut microbiota has been interesting for a long time, but we haven't really seen a lot that has talked about the role of histamine in gut bacteria, because it wasn't really traditionally thought that gut bacteria could produce histamine until we started finding evidence of HDC in these bacteria in a list you can see over here on the right. And so all I'll say about this for now is that it's quite relevant to inflammatory bowel disease, because these are patients who are not quite atopic, but are certainly related to histamine receptor dysfunction, especially having lower levels of H2 signaling, which is supposed to be that sort of immunosuppressive histamine receptor. So it's a potential explanation for why inflammation is sort of running rampant in IBD patients. And in the next section, we'll talk a little bit about H1 antihistamines.